Welcome back to the second part of the NCERT reading series of the chapter Principle of Inheritance and Variation. 4.3 Inheritance of two genes. Mendel also worked with and crossed pea plants that differed in two characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and one that had seeds of green color and wrinkled shape. Figure 4.7. Mendel found that the seeds resulting from the crossing of the parents had yellow colored and round shaped seeds. Here you can tell which of the characters in the pairs yellow, green color and round wrinkled shape was dominant. Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use the genotypic symbols Y for dominant yellow seed color and small y for recessive green seed color, capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital R, capital R, capital Y, and small r, small y, small y. The cross between the two plants can be written down as in figure 4.7, showing the genotypes of the parent plants. The gametes capital R, capital Y and small y unite on fertilization to produce the F1 hybrid capital R small y, capital R small r, capital Y small y. When Mendel self-hybridized the F1 plants, he found that 3/4 of F2 plants had yellow seeds and 1/4 had green. The yellow and green color segregated into a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. 4.3.1 Law of Independent Assortment. In the dihybrid cross, figure 4.7, the phenotypes round yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green with 3 round is to 1 wrinkled green. This derivation can be written as follows: 3 round, 1 wrinkled, 3 yellow, 1 green equals to 9 round yellow is to 3 wrinkled yellow is to 3 round green is to 1 wrinkled green. Based upon such observations on dihybrid crosses between plants differing in two traits, Mendel proposed a second set of generalizations that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of the two pairs of genes during meiosis and the production of eggs and pollen in the F1 capital R small y, capital Y small y, capital R small r, capital Y small y plant. Consider the segregation of one pair of genes capital R and small r. 50% of the gametes have the gene capital R and the other 50% have small r. Now, besides each gamete having either capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% of capital R and 50% of small r is independent from the segregation of 50% of capital Y and 50% of small y. Therefore, 50% of the R-bearing gametes has capital Y and the other 50% has small y. Similarly, 50% of the capital R-bearing gametes has capital Y and the other 50% has small y. Thus, there are 4 genotypes of gametes, 4 types of pollen and 4 types of eggs. The 4 types are capital R, capital Y, capital R, small r, capital Y, and small r, small y, each with frequency of 25% of 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollen on the two sides of the Punnett square, it is very easy to derive a composition of zygotes that give rise to the F2 plants, figure 4.7. Although there are 16 squares, how many different types of genotypes and phenotypes are found? Note them down in the format given. Can you, using the Punnett square data, work out the genotypic ratio at the F2 stage and fill in the format given? Is the genotypic ratio also 9:3:3:1? 4.3.2 Chromosomal Theory of Inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, his concept of genes or factors in Mendel's words as stable and discrete units that control the expression of traits and of the pair of alleles which do not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, through Mendel's work suggested that factors, genes were discrete units. He could not provide any physical proof for the existence of factors or say what they were made of. In 1900, three scientists, De Vries, Correns, and von Sandmark, independently discovered Mendel's result on the inheritance of characters. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structures in the nucleus that appeared to double and divide just before the cell division. These were called chromosomes, colored bodies, as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement, figure 4.8, to explain Mendel's law, table 4.3. Recall that you have studied the behavior of chromosomes during mitosis, equational division, and during meiosis, reduction division. The important thing to remember are that chromosomes as well as genes occur in pairs. The total alleles of the gene pair are located on homologous sites on homologous chromosomes. Table 4.3 A comparison between the behavior of chromosomes and genes. A occurs in pairs. B occurs in pairs. A segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. B segregate at gamete formation and only one of each pair is transmitted to a gamete. A independent pairs segregate independently of each other. B one pair segregates independently of another pair. Can you tell which of these columns A or B represent the chromosome and which represent the gene? How did you decide? During anaphase of meiosis 1, the two chromosome pairs can align at the metaphase plate independently of each other, figure 4.9. To understand this, compare the chromosome of four different color in the left and right columns. In the left column, possibility 1, orange and green is segregating together, but in the right hand column, possibility 2, the orange chromosome is segregating with the red chromosome. Possibility 1, one long or orange and short green chromosome and long yellow and short red chromosome at the same pole. Possibility 2, one long orange and short red chromosome and long yellow and short green chromosome at the same pole. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principle and called it the chromosomal theory of inheritance. Following this synthesis of Ideas, Experimental Verification of the Chromosomal Theory of Inheritance by Thomas Hunt Morgan and his colleagues, led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies Drosophila melanogaster, figure 4.10, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscope. 4.3.3 Linkage and recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow bodied, white eyed females to brown bodied, red eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other and the F2 ratio deviated very significantly from the 9:3:3:1 ratio expected when the two genes are independent. Morgan has his group knew that the genes were located on the X chromosome section 4.4 and so quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined the term linkage to describe the physical association of genes on a chromosome and the term recombination to describe the generation of non-parental gene combinations. Figure 4.11. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, showed very low recombination figure 4.11 cross A, while others were loosely linked, showed higher recombination figure 4.11 cross B. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Sturtevant used the frequency of recombinations between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genome, as was done in the case of the human genome sequencing project described later. 4.4 Polygenic inheritance. Mendel's studies mainly described those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short people as two distinct alternatives but a whole range of possible heights. Such traits are generally controlled by three or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele, that is, the effect of each allele is additive. To understand this better, let us assume that three genes A, B, C control skin color in human, with the dominant forms A, B, and C responsible for dark skin color and the recessive forms small a, small b, and small c for light skin color. The genotype with all the dominant alleles capital A, capital A, capital B, capital B, capital C will have the darkest skin color, and that with all the recessive alleles small a, small b, small b, small c will have the lightest skin color. As expected, the genotype with 3 dominant alleles and 3 recessive alleles will have an intermediate skin color, and in this manner the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. 4.5 Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. So that's it for today. If this helped you, then please support the channel and stay tuned for the next part. Thank you for watching.